Okay, so I'm going to give a quick recap of yesterday, and then we'll go to our breakout sessions. So I think of prostate cancer as three big categories of people: the newly diagnosed, the people that have a relapse. Do we have to turn all the lights out? I think we'll go back to sleep. Thank you. The people who have PSA relapses after surgery or radiation, and then the people that have more advanced or metastatic or hormone-resistant disease. And yesterday we heard talks that addressed each of these three areas. These are all very big areas, as we know, 200,000 newly diagnosed every year. And the relapse is about around a third of men that have surgery or radiation relapse. And then they have a very, very long survival. So there's probably over 1 million men in the United States that have relapsed prostate cancer. And then for some of those, they'll evolve into hormone resistance. So just some comments on the things, and I've got a few slides to show you as well. One was that Dr. Strum presented the latest data from Dr. Klotz on active surveillance, which showed, the bottom line was 450 men watched 10 years, and 5 men of the 450 that passed away from prostate cancer during that 10-year period. So there were a few people that lost their lives, but relative to the number of men that were able to avoid treatment, it was obviously this is something that needs to be discussed with patients as a viable option. I think that's the bottom line of the active surveillance situation. It's not a matter of if, but more a matter of how. What's the best way to do it? Should people have scans? How often should they have biopsies? And these are details that are being worked out and will no doubt be discussed at the active surveillance session today. Dr. Ornish, I can't add, I was awed at his ability to talk about how important it is we interact with each other and justify it scientifically. I didn't think that was possible. So that was incredible. There are studies showing the power of support group involvement, and we all know intrinsically in our hearts that it's good to be able to relate with each other, but I didn't know that it actually had been proven scientifically. I have one slide I'm going to show on Dr. Roach's presentation because I thought he packed about 7 talks into one talk, and it was all good and all important. I spoke briefly. I just wanted to remind you, because I was the only one that talked on this group of men with PSA relapse disease, that, and this has historically just been managed by hormone blockade. There is some controversy about intermittent hormone blockade, although that's fading away. It's becoming fairly widely accepted now. And so building on that, what were the methods to keep men on holiday longer? And we talked about using these types of medicines because they have a better quality of life, basically less side effects. And then the third section, this is where a lot of the research is for obvious reasons. These are the people that are facing possible mortality from the disease. And these people are selected for the trials because the pharmaceutical companies have been forced by the FDA to show a survival advantage. And I'm going to go into that a little bit more in my slides. But it's the distortions of what the FDA requires for FDA approval that creates all the studies being done in this group. Because if you do survival studies in this group or this group, it would take 15 to 20 years to do such a study. And most drugs, the patents run out after 17 years. So it's just a practicality. And of course, no one's really arguing about it because we know we need solutions for these people. So this is a summary. This is a slide that Dr. Roach presented yesterday, and I'm just going to go back and re-answer the questions that he presented such convincing conclusions for. Is radiotherapy as effective as surgery? The answer was yes, it is. And last year, Dr. John Blaskow was here and showed data comparing surgery and radiation, and his conclusion was that the radiation was better than surgery. And we're talking cure rates now. We're not talking quality of life. That's a separate and important question. So the cure rates for people with radiation are at least as good as surgery. And I'm convinced by the data that the cure rates are probably a little better with radiation than surgery. So, and then the side effects he didn't talk about as much, but pretty much universally there's been a number of studies published on quality of life showing better quality of life with radiation options than surgery. As you know, prostate cancer is the only remaining solid tumor that is run by surgeons. 30 years ago, all the cancers were managed by surgeons. Surgery was the only effective treatment for cancer 30 years ago. And urologists as a specialty group have existed for close to 100 years. And the medical oncology profession has penetrated the management of all the other cancer types except for early stage prostate cancer. And to this day, early stage prostate cancer is managed by surgeons. And in the training programs like at USC or UCLA, there is no training in early stage prostate cancer for medical oncologists. That's why there's so few of them in this area throughout the country. The second question he addressed was, is there scientific support for giving hormones with radiation? And I think it's important to emphasize that he discusses intermediate and high-risk prostate cancer. The PCRI has a brochure called What's Your Type? And I contend, and the PCRI contends, that if you don't first define the type of prostate cancer you're talking about, you're just going to be totally confused. Because if you have a low-risk type, you shouldn't be treated the way as a man with either intermediate and high risk. In fact, the general feeling now is that low-risk people can be safely monitored on active surveillance. So he's very specific and precise that he's talking about treatment for men with intermediate or high-risk prostate cancer. And in the studies that he presented, was compelling evidence that a short course of hormone treatment improved long-term cure rates with the radiation. Again, he didn't address quality of life. We know there's more side effects, and the cure rates, particularly with intermediate risk disease, are getting to be so good with radiation alone that there's certainly room for saying, well, if I give up a couple percentage points of cure rate likelihood, that maybe that's justified so that I can avoid the side effects of the hormone blockade. That's something that has to be discussed on an individual basis. But if you look at just cure rates and ignore side effects, it appears to be quite true that adding a little hormone therapy to the radiation will enhance the cure rates. And it's, I would say, a foregone conclusion with high-risk men that they need to get hormone therapy plus radiation. The third question he raised was, is cardiac toxicity a major concern? Of course it's a concern. The question is it a risk? I mean, we know that men tend to gain weight and with this treatment if they're not careful. And what does weight gain cause? It causes higher blood pressure, it causes diabetes. So yeah, there's going to be secondary heart effects in people that are not prudent in how they manage their diet and keep their weight under control. But how big a problem is it? It's a lot smaller problem than the number of men that are getting improved survival by adding the hormone treatment to the radiation. So there is a cost of hormone therapy, both in quality of life, and then if they're not careful, if people do get diabetes, hypertension from their weight gain, it's going to be, inevitably, we already know that there's going to be an increased risk of heart issues. So it's just a relative factor, though, and he pointed that out very nicely. There are some, then the last question he addressed was this controversy about whether or not we can radiate the lymph nodes in the pelvis and whether that is advantageous. This was, I think, a difficult question 10 years ago when we didn't have safe radiation for radiating the pelvis. Men would get their intestines burned all the time, and that was very, very discouraging. Now that we've developed IMRT and IGRT, I personally have had at least 30 or 40 patients go through some form of lymph node radiation, and so far have not seen a single case of notable bowel toxicity. And this is because of the moderation they can shoot around the intestines. So even without the evidence that Dr. Roach presented, which I think is interesting although not conclusive, I think that logically speaking, from what we know about all the management of all other cancers, that if you can control the disease in the lymph nodes, that you're going to get better results as long as we're not inducing a lot of unnecessary harm. So I thought these were really important questions that Dr. Roach addressed, and he did it very adroitly. A lot of complex slides and graphs and numbers and whatnot, but this was the take-home message from that, and very important ones indeed. So I'm just going to fill in a couple gaps I thought for advanced disease that there's a lot of new things coming along. And I think that's one of the messages that you want to take home from this conference, is that we're in a period of exponential growth for the development of new treatments and research. And this is a very hopeful thing, that since the beginning of time, there was very little advance. In the last 100 years, we've seen some medical advances. In the last 20 years, a little more. In the last 5 years, just about as much as we probably saw in the last 20. And then in the next 5 years, we'll probably see more than we've seen since the beginning of time. And this is because, as Jonathan Simons pointed out yesterday, they're starting to figure out what cancer is so they can design specific solutions. When I started on oncology 20 years ago, it was a situation of them giving poisons to petri dishes randomly. They would try things like car wax and rat hair and all sorts of things. And if the cancer cells died, they didn't know why, they would feed it to a living animal with cancer. And if the cancer in the animal died, and the animal didn't die, then they gave it to humans. And that's how we developed chemotherapy. And that's why most chemotherapy has got this bad reputation of terrible toxicity, because the chemotherapies just work by inhibiting the growth of all cells. So growing things like our hair, the lining of our mouth, all these things would get hit in the crossfire from these treatments. Now we're dealing with new treatments that pinpoint what is different about the cancer. And that's why we're going to see much more effective treatments, and we're going to see far less toxic treatments. So, for example, this is what was FDA approved in prostate cancer as of the 2000s. One medicine in the 80s, one in the 90s, and one in the last 10 years. And this has become fairly standard. And then we look at just 2010 alone. Provenge approved, Cabazitaxel. And because of that press release yesterday about abiraterone is I call it a safe type of ketoconazole. Dr. Myers gave a long talk on ketoconazole yesterday, and he bemoaned the fact that it's such an effective drug, but so difficult to use. And there aren't very many medical oncologists treating prostate cancer, and the urologists are intimidated by something that could have toxicity approaching that of chemotherapy. Well, abiraterone is going to solve that, because this is extremely well tolerated. We treated 27 people in our practice on the phase 3 trial, and we didn't have to stop the medicine in anybody. We saw essentially no problems, and it is, I feel, at least as active as ketoconazole. So this is very exciting, and when the FDA sees a survival advantage, which is we're going to go into in a minute here, that is they pretty much almost have to approve it. That's what they asked for. That's the way they told these pharmaceutical companies to run the game. So that if they play the game by the rules, and people live longer, because in the abiraterone trial they treated half the men with abiraterone, half of them with a placebo, and that was the press release on Friday that the company reports that the men on the abiraterone did indeed live significantly longer. So that's the Cabazitaxel. They announced that survival advantage sometime in May. The FDA approved it like in June or July. I can't remember the specifics. So that's what they asked for. That's what they're going to get. So it looks like we're going to get abiraterone pretty quick, assuming that Johnson & Johnson can manufacture enough to keep up with the demand. So I want to talk just a bit. When we come to a meeting like this, you get a lot of scientific talks, and those talks are presented in doctor speak and lingo, and this was addressed briefly yesterday, but I wanted to readdress this question of survival advantage, which is the type of study demanded by the FDA, but may not relate that well or be that understandable to patients. So, you know the old idea, half the people get one treatment, half get the other. They make this decision by a coin flip to even out the irregularities, and then they watch which group, on average, lives longer. And so this is a classical FDA type design, and this is a Taxotere study that got Taxotere approved, that there was a couple months difference in survival, but statistically significant. In other words, the chances that this difference could have happened was less than 9 chances in 1,000. So they said, this is probably for real. There's probably really an effect there. But no one gets very impressed by the effect. So the trade-off, of course, is that Taxotere has side effects. And when people hear this number of 3 or 4 months, and then they look at the fact that it could cause hair loss, could make them feel tired. They say, why would I want to spend my limited time being miserable? And I think I'll pass. So this, again, is because of the design. The FDA just designs a horse race, and if the new medicine wins the race and the placebo loses the race, they figure, okay, there's activity, we'll approve it. And there's reasons to argue for and against this style of testing, which we're not going to go into, but that's just the way the FDA has made it happen. So, for example, if you have someone that goes on a treatment and has cancer spread all through their bones, and then they get on the treatment and it all goes away, the FDA is not impressed by that. That's just that's not enough. They want to see big groups of people, median survivals different, period. So this is why you keep hearing these survival studies being done. But what people are forgetting is that just because the average of one group is only a little bit better than the average of the other group, the individuals in the trial, there's a huge variation, and there's a way to tell as the process unfolds whether you're getting benefit early on or not. This is a redrawing of the same curves of that study showing that the men that had at least a 30% decline in PSA had a much bigger separation of the curves. And if you took away the men that didn't have a 30% decline in their PSA, they were almost the same. So how long does it take to find out if you've got a 30% decline in PSA from Taxotere? About 60 days. That's usually what I tell patients. So you'll be able to discover quite promptly, two treatments, maybe 3 at the most, is this stuff going to work for you or not? And if not, stop it and look for something else or an alternative. So this is what really has to be emphasized, is when you take the people who really do benefit, which is not everybody, the benefit is much bigger than a 2 to 3 month benefit. In fact, if you look at the difference in the curves in people that were able to get their PSA all the way down into the normal range, these are people that had a PSA drop to less than 4, you can see that the difference in survival, these are the survival curve of the people that had their PSA drop to less than 4. It's the same study, Taxotere versus mitoxantrone, versus the people who didn't get their PSA down to less than 4. And what is that? That's a 20-month difference in survival, averaged. Some people did better than 20 months, some did worse than 20 months, but average improvement in survival. So again, 20 months is not a huge number either, but it is certainly a significant thing to consider, especially in light of the fact that the slides that we're going to go into now are that we can enhance the results with Taxotere. There are Taxotere plus protocols that are better. One thing that we need to remember, that's just PSA. In situations where there's ambiguity about who's responding and who's not responding, there are other tests, many other tests that can be done at baseline and then monitored, just like PSA is monitored. It should go down if the treatment is working, and it goes up if the treatment's not working. So a number of blood tests that hopefully you've heard about, circulating tumor cells, which were mentioned yesterday. This is commercially available. Bone scans, and how people feel. If people are having any soreness or pain from cancer, that's the first sign of a response usually is that the pain goes away. That can happen within a couple weeks of starting a treatment. So this is all addressing the issue of treating people like individuals, not like a herd of sheep, and you're just one of the sheep in a big herd. I want to just quickly go through some things that weren't mentioned yesterday, maybe because they're not on trials, but they've been around for a while, and I think there are studies showing that they are indeed effective and they make Taxotere work better. One is the medicines that are derived from a type of chemotherapy with a platinum component in it. So here's just a few trials. It's mainly carboplatin, which is one of the milder platinum medicines, that shows that if you have this, of course, these people were heavily pretreated, and they didn't get too good a result. And also there was no MSET. But when you use carboplatin and MSET together, whereas there's about a 50% response rate with Taxotere alone, response rates go up to quite a bit better. This is another platinum derivative that's approved in colon cancer So we haven't been able to get insurance coverage, but these people were heavily pretreated with 2 other chemos before they went on this, and they still 2/3 of the people responded. So there is definitely activity by adding either platinum or platinum NSAID along with the Taxotere. The other medicine is a pill that's approved for breast cancer called Xeloda, and it is also quite active. You can see that if you add Taxotere and Xeloda, that if you say average response rates are about 50% with Taxotere alone, that you're getting higher response rates with this. So we see this in the clinic where men come to the office and they've already had Taxotere and they've been told it's not working, the PSA was rising, and then we'll reinitiate Taxotere either with Xeloda or carboplatin, and then they will respond. So we know that even though they were resistant to Taxotere, sometimes they'll respond to the combination of Taxotere with something else. This is a new medicine that's coming that's just kind of exciting. It's still only in phase 3 trials. This trial will be opening up, I think, in a month or so. But there's a few unique things about this. I'll see if I can say this without... It's called an antisense oligonucleotide, if you catch that. Antisense oligonucleotide. Does that explain it for you? In fact, practically all the medicines that you hear about are designed to block proteins and enzymes, things that have already been structurally created by the body to fulfill a certain purpose. The oligonucleotides, we're talking more at the DNA level or the RNA level. That's like the information level rather than it's like if you have building plans versus a house. So all the treatments that are used for the treatment of cancers are usually designed to burning down the house. This is a medicine that's designed to changing the plans so that the house doesn't get built or gets built differently. And so if this pans out, obviously this is going to be very, very exciting because they can go in and they can alter all kinds of genes this way. In this case, they selected a gene that makes a protein that breaks down Taxotere. So when we give Taxotere, it goes into our bodies and gets into the cancer cells, kills some of them, but there's a protein in the cancer cells that starts to gobble up the Taxotere and get rid of it. And that protein is called clusterin. So some genius thought of the idea of, well, let's just erase clusterin from the building plans so that there is no more clusterin. And so this has been developed, and these are sort of some complex cartoons that explain all the building plan concepts. I don't think we'll go through those. Maybe we can just briefly, quercetin, which is the name of the medicine, is a string of DNA it's actually a string of RNA. So here's DNA, probably most of you recognize that. And then RNA is made from that, and then it gets transported out into the cell, other part of the cell, and then from their RNA they make the proteins. These are the house, so to speak. So this is the information, the plan, and this is the house. So what they've done is this clusterin protein that the cancer cells are making in their DNA, on its way out to get turned into a house, they make this quercetin stuff that acts sort of like glue to stick to this MRA, this building plan that's on its way to the building site, so that the building plan never gets to the building site. So you see it here sticking together with here's the medicine that they made, here's the RNA that's on its way to the building site. They stick together and therefore inactivate it. So does it work? Well, they have one small randomized prospective phase 3 trial in 80 patients that showed a little bit better PSA response when they added the quercetin, I don't know if I pronounced it, and versus Taxotere. Any drop in PSA, definitely a difference, but one of the biggest differences in survival that we've seen. So look at that. It's not. This is a really big difference. So that was intriguing enough that the pharmaceutical company is ramping up to start a phase 3 trial of hundreds of patients, so looking towards FDA approval, and that should be available in the next few months or maybe even next month. the side effects of this added substance did not seem to be an issue. So this shows the survival curves. You're getting used to seeing those now. Okay, so just to review then, things that seem to make Taxotere better: carboplatin and MCET, Capecitabine, otherwise known as Xeloda. There, quercetin we talked about. Xeloda, I didn't talk about. That's just finishing phase 3 trials, very well-tolerated medicine that may make Taxotere better. We may hear about that in the next, 3 to 6 months. And then Revlimid is another trial that's opening up with and without Taxotere. Probably going to help. We talked a little bit about Revlimid yesterday. And then there's a number of things that have been looked at which probably aren't helping very much. It's also good to know those, so that if someone's got a doctor who's a little out of date, and I'm not saying we never use these, but we certainly would try these first. These are not inactive, they're just statistically less likely to work, and if you're going to start on something, you want to pick the one that has the best chance for success. So this was also announced by Dr. Vogelsang yesterday, that they've developed a new improved Taxotere. And the reason we know it's improved from Taxotere is they took men that were resistant to Taxotere, 750 men who had all been treated with Taxotere, and the Taxotere was proven to have stopped working. Tough group to treat. Pretty gutsy trial for a company. I don't know what they gambled on this, $30, $50 million, something like that. They must have had a lot of confidence that this is going to work. Typical FDA-designed trial. I mentioned before that once this trial was shown to be positive, the FDA approved it within just a month or two, and you could see that there was clear separation in the survival. This thing has activity in people who are Taxotere resistant. As you all know, us oncologists get excited about this because we think, well, if it works this well in people that have resistant disease, is it going to work a lot better in people who've never had Taxotere? And how much better will it work if we combine it with some of those other known active agents like platinums or Xeloda or the quercetin? So these medicines then can be used in combination, used earlier, and they have an even bigger impact. So I already showed you this slide. Just to review, though, that there's people need to be treated as individuals, and to do that you need to monitor the individual closely, check the PSA monthly, along with some other tests. And then when something's not working, change the treatment. So I think that's it for our talk this morning. I would like to just re-announce to you that you need to fill out some question cards for the upcoming roundtable, which will be after the breakout sessions. And don't forget to complete those evaluations and drop them in the foyer. And I think we're meeting back here around 11 or so, I'm not sure, but it'll be after the breakout sessions. So we'll see you a little later.